voles. The female voles don't have the big asymmetry. The two species of voles don't have the big asymmetry in their behavior to start with, apparently. So yeah, I think it's a guy thing. More on that later in the term. Yes? Yeah, reading brain signals, we'll call that recording in some fashion or other. If you can stick an electrode into the brain and stimulate, you can also stick a electrode into the brain and record from brain tissue and ask what a particular cell or bunch of cells are doing. You can do this at a large scale level in walking around human beings or animals by putting electrodes on the skull. You can read massed activities of large numbers of neurons off the surface of the skull. That's a so-called electroencephalogram, EEG. And that's very useful, for instance, in telling the difference between different wake and sleep states. If you are deeply asleep, the sort of sleep that when the alarm clock goes off, you're, where am I? If you look at the EEG, you find big, slow waves. All the cells are active, and then all the cells are quiet, and they're all firing together. When you're looking at an awake individual, the waveform is much smaller amplitude and higher frequency because the calculus piece of your brain is currently napping, and the psychology piece is wide awake, and various bits are doing things out of synchrony with each other. So you can see that from massed recordings. But if you now go and stick an electrode, let's suppose we go and stick an electrode into, let's take this same cell that stimulate here, and the patient said, I feel that in my toe. If instead of stimulating you record, you find out that cell doesn't change its behavior at all over a huge range of possible things that the organism could be doing. But if you poke the toe, that cell says, here I am. I'm interested in that and exactly that. You go poke the armpit or something, the cell doesn't care. You poke the other, the next toe over, the cell doesn't care. You poke one particular toe, let us say, that cell will care. Take a cell out here somewhere, and that cell will care only if you put visual stimuli in a particular little spot in the right side of visual space, in a very particular part of visual space. Move over a little bit, and it'll want stimulation in a different piece of visual space. Move elsewhere in the brain, and now, rather than liking, say, just something simple like a line moving around, a cell down here, this is typically done in things like monkeys, and so that cell might be very interested if you show it the hand of a monkey. Right? Cell's thrilled. Show it, a hunk of chalk. I don't care. Nearby one might like the face of a monkey, and so on. So you can find out what individual cells are interested in by recording their activity. There are some significant drawbacks to this, which is that one of them is that you can only sample from a tiny fraction of the cells, even in an area that you are interested in. How many cells do you think how many neurons do you think there are in the brain? So the answer is, yes, a lot is a good answer. But the answer is going to be expressed in terms of 10 to the x. What's x, would you guess? The 24s are a little high and the 9s are a little low. When I asked this question when I was in graduate school, I got the answer that there are nobody knows the answer for sure because who's going to go and count? That there are 10 to the 12th neurons in the brain, and of these, I didn't even tell you on this picture, but here we can draw a tail on this thing. Hanging off the back of the brain, sort of underneath the back, is the cerebellum. It means little brain. Important in learning and motor control sorts of things. We're not going to say much about it here, sadly, but what my professor said was there are 10 to the 12th neurons in the brain. Of these, 10 to the 13th are in the cerebellum. The quick on the uptake crowd picked up that there's a problem here, right? What he was trying to tell me was not that he didn't understand math, but that, look, we don't even really know to an order of magnitude how many cells there are in the brain. But that lots on this order. How many cells can you record from in a study of some function in the brain? You know, heroic single unit, single cell recording studies might have, you know, on the order of 10 to the second, 100 cells recorded from. Moreover, if you're going to stick an electrode in the brain, the cells that it encounters are going to be biased by things like size. You're more likely to get big cells than little cells, and big cells do things that are different than little cells do in the brain. So it's not, you can imagine that it would be difficult to figure out what your laptop did by recording from single components on the motherboard. That wouldn't be particularly easy. It gets significantly worse when you try to do that in the brain as a whole. The things we know best about in the brain are things where we can bring all of these techniques to bear and get what would be called converging evidence, converging data on it, evidence from multiple different sources, and then corroborate that by looking at behavioral measures. Oh, let me say, well, all right, this is the moment in the lecture where my wife, where I really feel my wife looking over my shoulder. In 2 minutes, I will polish off the rest of the brain. So that's just the surface of the brain, right? You know, the brain is a 3D structure. One way to very grossly think about the overall structure of the brain is that if there's cortex, that lying underneath cortex is what are a bunch of structures that often get called limbic structures. This is really an outdated term. It comes from the word for ring, and it was the notion that there was a circuit of structures lying under the brain, under the cortex. But there's a collection of structures who will show up later in the term with names like the amygdala, from the Latin for almond, hippocampus. Is it Latin for almond or Greek for almond? I'm not sure at the moment. Hippocampus, from the word for seahorse. Not because you have almonds or seahorses in your brain, but because the shapes of these things are faintly reminiscent of such things out in the real world. And other structures that turn out to be vitally important in emotional, in your emotional life and in memory. Hippocampus will show up later as critical in memory. Lying underneath there and heading on down into spinal cord, I don't know, let's call it the core today or something, are a set of structures that, well, we tend to spend a lot of time in psychology talking about the cool stuff happening up in the cortex and stuff like that. But the stuff you really don't want to lose, the stuff we don't know about from lesion studies, is down here. Because something in your brain has to say to your heart, beat, beat, beat. That piece of brain goes, you got really big trouble. There's another piece down there that says, time to breathe again. I mean, it's not glamorous work, but, you know, somebody... So there's a whole set of stuff here running vegetative processes, and also important, not just for, you know, pacemaker kinds of things, but also for general issues of arousal and sleep and wakefulness, those sort of things. So that is an embarrassingly brief tour of the... You can spend your life on neuroanatomy. Trust me, my wife does. We'll come back and talk about single cells on Tuesday.